Welcome to another lecture. This one is on membrane transport. Okay, so what you see in this figure is a number of different ways to transport molecules from the outside to the inside, or vice versa, to a membrane. So you've probably heard about this in cell biology, maybe even a little bit in Bio 111. There are multiple ways how cells achieve the transport of these molecules through a membrane that otherwise would not allow this molecule to penetrate the lipid bilayer. So there are, for example, these channel proteins, which are an example of simple diffusion. So in simple diffusion, the molecule moves from one direction to the other direction, depending on its concentration gradient. So there has to be more on this side than on this side in order for it to move either directly through the membrane, if it can do that. If it cannot do that because it might be too charged or it might be too large, then it can move through a channel protein that will allow it to pass through the membrane, but only along its concentration gradient. So that's also referred to as passive transport. There are carrier proteins that are different from channel proteins in that they take the molecule and then they change the conformation of the protein, and that allows them to pass through the membrane. But again, carrier proteins also only work with respect to the concentration gradient. So there has to be a concentration gradient between the two compartments here from this side to that side in order for it to move. If a cell wants to move a molecule against the concentration gradient, it requires energy. And so for that, it requires something that's called active transport as opposed to passive transport. With active transport, you use energy such as ATP in order to produce a gradient. So you can actually start a gradient in the other direction by pumping actively using ATP molecules from one side against their concentration gradient to the other. So when you do any of these transport possibilities here, you can change the concentration of one side versus the other side. And if the molecule is an ion and is charged, then that will also create potentially an electrochemical gradient because you are now accumulating charges on one side versus the other side. So that's sort of the overview of membrane transport. So it's always a little bit tricky to remember how to describe a membrane transporter. And one way that I find easier to remember than just all the different possibilities is that there are three ways how you can describe a transporter. And any one transporter can be described in these three ways. So you can sort of mix and match, if you will, with how you describe this particular protein. So first of all, you can always say, well, is it an active or is it passive transport? So is it active, then ATP is used, or is it passive transport, then no ATP is used. And then the question is it primary transport or is it secondary transport? So in primary transport, you have one protein that transports and provides energy if that is required. In secondary transport, you have the energy uncoupled from the transporting. So the energy for the transporter comes from a different protein, for example, a hydrogen ion pump that sets up a gradient that is required then later on for that other molecule to be transported in the secondary transporter. So primary transporter sets up the gradient that the secondary transporter uses in order to move a molecule through the membrane. So you have active versus passive, you have primary versus secondary, and then you also have antiport versus symport. So an antiport carrier is one that uses two molecules, where two molecules pass through the transporter in opposite direction. So as one is moving in, the other one is moving out. And so therefore the gradient for that one molecule that's passing in one direction can be used to bring in a molecule from the other direction maybe even against its gradient. So it can use the energy from those molecules from the antiport in order to bring in a molecule into from the opposite direction. And a symporter is very similar to an antiporter, except that they're not going through a revolving door, one in, one out, but both going in at the same time or both going out at the same time. So there you have two molecules that pass the transporter in the same direction. We'll take a look at a number of those examples as the lecture progresses. Okay, so there's a few more things, things we need to discuss before I can show you some of these examples. So ATPases, that is the name of a particular type of transporter. ATPases are hydrogen pumps, and these hydrogen pumps create a gradient of charges, of charged hydrogen ions. That is also sometimes referred to as the proton motive force. Protons, because these are protons, right? Hydrogen ions are protons, and they have a force that allows them to move things around, either themselves or through secondary transport, other molecules, ions, and so forth. So ATPases are very important because they create a charge differential and a hydrogen differential that can be used for a number of different transport ions, not just for one particular type. You'll see that as well in a little bit. So as hydrogen ions get pumped in one direction, you get a charge buildup, and that is also referred to as the electric membrane potential. And we'll see that the membrane potential across a membrane, so the charge that results from negative or positive charges inside versus outside two membranes can be different. And if they are different from each other on those two sides, then you have created an electric membrane potential. And such electric membrane potential can be powerful for the plant cell or for the animal cell, as that may be, but we're talking about plant cells here, with a concentration difference as little as 0.001% on the outside versus the inside of a particular molecule, let's say hydrogen ions, that can create a difference of 100 millivolts between the two sides. So building up a relatively strong membrane potential, as you will see in a second, doesn't require a huge amount of protons. All right, so let's sum up a few things that I said at the beginning. When I showed you the slide with a picture of those three different types. I talked about channel proteins. So channel proteins are tunnels that transverse the lipid bilayer. They are not open all the time. They are gated. So gating is the term for molecules that can operate these gates in order to open the channel proteins. And these molecules that operate these gated channel proteins are sometimes hormones. They can be voltage, so that would be a voltage gated channel. They can be light signals. They can be proteins, enzymes such as kinases or phosphatases that will activate a certain channel protein. And then also you can refer to these channels as channels that go inward inside to a cell or outward. And so they're called inward rectifying or outward rectifying. So that's channel proteins. That's something you might want to remember about the channel proteins. As opposed to the carriers, remember that we looked at those at the very beginning as well. Carriers do not traverse the entire membrane, and the transport is much slower than through a channel. So the transport there is accomplished by change in the conformation of the protein. So something changes inside the protein that allows the to be transported molecule to be pushed through the membrane and then sort of expelled on the other side. And many of the pumps carry out energy costing, active transport, and that energy is provided by ATP hydrolysis, or, and I hadn't mentioned that so far, by redox reactions. So they can also occur in order for them to provide the energy to turn on these pumps to create a gradient. All right, so lots of different definitions. Let's take a look at some of these membrane proteins, and then let's take a look at some of their functions and what they do for the plant. And then all of this will culminate in the discussion that we'll have in class, where we discuss the paper by Gaxiola et al. About salt transport and salt responses in plants. All right, so here's a cell, and here are all the different types of cell transporters, plasma membrane transporters that a particular cell can have. So not every cell will have all of them in the same amount, but these are all the ones that are known. And I'm pointing out here a cell with a plasma membrane on the outside and the cytosol here. But remember that plants also have a vacuole, right? So the vacuole inside the plant cell is very important for a number of reasons that we'll discuss in more detail. Vacuoles also have a membrane. So you have a vacuolar membrane here, and that membrane, just as the plasma membrane, can harbor a number of integral proteins and transport proteins, for example. All right, so if we take a look here at the top, they are just sorted so that they fit into these nice categories, right? They're certainly not sorted in this arrangement in a cell membrane. So these are all symporters. You can see that something goes in, something else goes in at the same time. So you have, for example, a potassium, sodium symporter, or a nitrate hydrogen symporter, as you have here. So if there is a gradient, a higher concentration of hydrogen on the outside, lower concentration of hydrogen on the inside, then that gradient can be used by this particular transporter, symporter, to take nitrate against its concentration into the cell. Right? So that would be one example. Here you have an antiporter, sodium out, hydrogen in. You have an efflux carrier for sucrose. You have a number of pumps, so those are all the yellow ones. So there's a hydrogen pump here. There's another ATPase pump. In this case, calcium is pumped. And then here we have more hydrogen pumps. There's one that requires ATP, and here's one that requires only the PPI, so the pyrophosphate. So that is the one that we will be discussing in our discussion of the paper. So these are across the tonoplast. That's the membrane that surrounds the vacuole. Here are a bunch of antiporters. Here are a number of channels. So you can look at all of those. This picture is also in your book. Just gives you an overview of all the different types of transporters that can be found in a plant cell. All right, let's take a look at an example. Let's take a look at how plants use these membrane transporters in order to survive a particular type of stress. And so the stress that we will discuss is salt stress. So if we just take a look at these plants, these are corn plants, maize, as they're often referred to both in England but also in plant biology in general. So you see these corn or maize plants. You have a control plant here. These were watered with just pure water, and then more and more salt was added to the watering solution. 50 millimolar, 100 millimolar, 150 millimolar, 200 millimolar of sodium chloride. And you can see that there's a drastic decrease in viability of these plants. 50 millimolar are still okay, but then you can see that the plants are much reduced in their growth, in their amount of chlorophyll, so that the amount of green pigment in their leaves. You see that there are many leaves that are dying, that are dried up, yellowing. Plant is much smaller. And if you were to take the plants out of these pots and look at the roots, you would also see that the roots are much longer here in the control and much shorter in progressively increasing concentrations of sodium chloride. So sodium chloride is sort of a double whammy for a plant. And it's because sodium is toxic. So the sodium ions are toxic for plants. Plants are much more sensitive to sodium than animals are, humans, for example. So humans actually require some sodium. Plants can do just fine without sodium, and any increase in sodium can cause stress. And that is because of two things. It's because of ionic stress and of osmotic stress. And so with the osmotic stress, we tie back into the water potential in a second, but let's just take a look here at the ionic stress. Essentially what that means is that sodium is toxic to the plant. So what I'm saying here is that the potassium-sodium ratio gets out of balance. So usually there's a ton more potassium than sodium in the plant. A little bit of sodium can be tolerated. Some plants need a little bit more sodium, but in most cases, potassium is required. But if sodium gets into the plant in a large amount, then this ratio is off. The next thing is that, well, there. So sodium is toxic to the plant. It causes leaf senescence. We saw that in the picture before. Leaf senescence means drying up, wilting, essentially. Sodium also has the ability to inhibit enzymes. So they start to change their conformation with too much sodium around. These plant enzymes are sensitive to sodium, to too much sodium around, just because they inhibit their function. And one of these examples is that photosynthesis is inhibited. Of course, photosynthesis relies on a number of enzymes, and if they are inhibited, then photosynthesis doesn't work properly. The plants no longer produce energy that they need for their metabolism, and wilting occurs, the plant dies. However, plants can also adjust to these conditions, and they can either adjust, the individuals can adjust somewhat, but some plants also, some species have also adapted over evolutionary time to higher salt concentrations in their surroundings. So there are salt sensitive plants that are very sensitive to salt, and some that are more resistant to plants. So there are even some plants that are called halophytes, halo meaning salt and phyte meaning loving or liking. So plants that like salt. I wouldn't really go that far that they really like the salt, but they have adapted, they have created strategies that will allow them to live in salty conditions. And we'll talk a little bit about that as well. So what they do, cellularly speaking, is they either excrete, so I'm talking about the plants that are halophytes that can withstand higher concentration of salts. They can excrete the sodium. They have, some of these species have certain adaptations that allows them to actually secrete salt out of their leaves in order to move it out of the cytoplasm, and in so doing then prevents enzyme inhibition. So they excrete the salt, or if they can't excrete it directly out of the cell, they can compartmentalize it within the cell. And I'll show you in a second, and we'll also read about that in the paper, how sodium can be compartmentalized into the vacuole. Sometimes people refer to the vacuole of plants as the trash can of the plant cell. And so you'll see how compartmentalization, moving away the sodium out of the cytoplasm, where many of these enzymatic reactions occur that the plant needs to do to fulfill its functions. If the sodium is compartmentalized away from the cytoplasm, then that will also allow the enzymes to be functional. So plants can excrete, they can compartmentalize. Some species have, they accumulate salt in certain leaves, and then they just drop the leaf off and create new leaves. So they compartmentalize salt within the body of the plant rather than just within the cell, within individual cells. So those are specific adaptations. The second part of that double whammy that I was talking about when it comes to salt stress is that salt stress also causes osmotic stress. So yes, sodium is toxic, but also sodium is an ion. And so with that, it's a solute, it creates a change in the solute potential, right? So if you have high concentrations of salts in the soil, and we talked about that in the previous lectures, then that will result in an inhibition of water uptake, right? So there is, it's more difficult for the plant to bring water into the plant because the water potential in the soil might approximate the same water potential in the plant. So then there's water stress. So salinity stress can then also lead to drought stress, water stress. And that is caused by osmotic stress, by the movement of water towards a compartment, be it the soil, for example, that is more salty and therefore attracts water. And of course, that creates problems with growth. Plants that grow in salty soils are smaller, and it's easy to see that they are not very happy in these osmotic conditions. But what you could do in order to change or to look at the ionic stress versus the osmotic stress separately is to take a non-toxic ion and compare that to a toxic ion such as sodium and see what the similarities are with respect to the responses. And you will see that the osmotic stress will also be felt by the plant if the ion itself is not as toxic to the plant. For example, if it was high potassium, you wouldn't get the ionic stress through the toxicity, but you would get the osmotic stress that that ion with the same charge will have on the plant metabolism. So cell responses that have been developed, that plants have adjusted to and evolved to deal with osmotic stress, is an osmotic adjustment and oftentimes an accumulation of non-toxic ions. So remember that any kind of ion or any kind of solute really can be used to lower the solute potential in a solution. So the accumulation of certain, for example, organic acids that have the same effect on lowering the solute potential as any other ion, for example, would have, allows the plant to sort of counteract the high concentration of solutes in the ground, in the soil. And so by producing more organic ions, for example, or organic acids that are in ionic form, or accumulating other non-toxic ions that can then, on the other hand, lower the solute potential again in the plant and allow the plant to take up water, even if there is a relatively low solute potential in the soil. So both of those strategies, all of these strategies can lead to recovery and adaptation, but they don't always do that, right? So it's not a given that a plant will be able to respond to acute salt stress by simply responding and then recovering. All right, so let's look at the cellular level of those types of membrane proteins that are involved in these kinds of adaptations or responses that I was just referring to. So here's a plant cell again, here's our vacuole. And so compared to the figure that I showed you at the beginning, where all those symporters and antiporters and transporters in general were shown, this only shows a selection of membrane transporters that are involved in the salt tolerance response. All right, so let's take a closer look. We have a membrane here, and we have a cell wall here, and then we have the vacuole here, and we have the vacuolar membrane. And then notice that the pH inside the vacuole is 5.5, and that the pH in the cytosol is 7.5. So that is a difference of what's the difference between those two values? Two, of course, right? But if you remember that the pH scale is logarithmic, then there's really a difference here in 100-fold difference in hydrogen concentration, right? In hydrogen ions. So there's a 100-fold difference between hydrogen in here versus in here, and then also in the apoplast, so on the outside in the cell wall outside the neighboring cells and so forth that are not inside the symplast but in the apoplast also hold a pH of 5.5. So you see that there's a membrane gradient between an electric gradient, that charge gradient, right? Because protons are charged. Well, you can see the difference here, 100-fold between the outside, the cytosol, and the vacuole. Now, how do you get this pH gradient to be maintained? Well, for that reason, we have the ATPases, right? So let's look at them. Here is one. So you see that ATP is broken down to ADP, and that is used to pump hydrogen into the vacuole. And so more hydrogen inside the vacuole, of course, means more acid in the vacuole, right? It's more acidic in the vacuole, and so the pH drops. Now, if the apoplast is also more acidic than the cytosol, there must also be a way for hydrogen to get out of the membrane, through the plasma membrane into the apoplast, and here is that ATPase for that. So ATP is broken down into ADP, hydrogen is moved out. And now you can see what the plant can do with this currency, if you will, of a hydrogen gradient. So hydrogen being pushed out here, hydrogen being pushed out here. Now it has a bunch of hydrogen in the vacuole and on the outside that it can use to do some of the work that it would like to do in order to maintain its enzyme activities that it requires. So here, for example, the hydrogen that was pushed out through this particular ATPase can now be used as an antiporter, right? This is an antiporter, hydrogen in, sodium out. And this one's called SOS1. That's just the name of the gene that makes this particular protein. And so therefore that protein is also called SOS1. The reason why it's called SOS1 is because that was a gene that was found in a mutant that didn't have this protein, and so that plant was particularly sensitive to salt, right? It couldn't do this sodium pumping out of the membrane. Couldn't do that because it didn't have this, and so that mutant was called a salt overly sensitive mutant, and so SOS, salt overly sensitive, was the name that was given to that plant that didn't have this particular protein. Anyway, let's come back to how hydrogen can be used as a currency to pump or to operate these pumps. Sodium out here. And then you see that there's another one here. Hydrogen out of the vacuole. You have a surplus of hydrogen here. The pH is low. pH is higher here. 100-fold. This NHX protein is also an antiporter. Sodium out of the cytoplasm, into the vacuole, into the garbage can of the cell, hydrogen out into the cytosol. And then there are a number of other channels and transporters that are involved in sodium regulation. And we'll take a look at that in a second. But just note, before it gets a busy slide here, that there are calcium transporters. There are some sodium channels. There's a calcium channel. There's a potassium channel. More calcium transporters that require some ATP. And here's the pyrophosphatase. That is also a hydrogen pump that doesn't require ATP itself, but remember that there is still energy left in the two phosphate bonds even after the triphosphate is split off, right? So it's the diphosphate. You can still split one more off and get more energy out of that in order to pump the hydrogen. All right, I told you it gets busy. So here is all the involvements that are activated in the salt tolerance response. So sodium itself, that the shape of the ion can activate by binding to this particular calcium transporter. And so when it activates that, then calcium goes in. So these are not, on all the time. I said earlier that some of these are gated. They have to be activated in order for them to become active. So sodium activates this particular transporter. Now, calcium moves into the cell, And when calcium moves into the cell, calcium is known as what's called a second messenger. It has information that it produces and that it gives on to other molecules, other proteins inside the cell. One of them is that it activates two other proteins of this family of salt overly sensitive plants. So there's protein SOS1, SOS2, and SOS3. And these are not membrane proteins. These are soluble proteins inside the cytoplasm, and calcium can activate them. When calcium activates SOS3, what SOS3 does is it inhibits, that's what this little bar means here. So an arrow is a positive activation, a bar is a negative activation or repression. So it represses any kind of transporters that still might allow sodium to get in, because apparently there's already too much sodium here. So let's make sure that all of these transporters are shut, and that's what SOS3 does. And SOS2 is a phosphatase. It's a kinase, and it uses phosphate in order to phosphorylate SOS1, which activates SOS1, which then starts pumping like mad all the sodium outside of the cell through the cytosol. So that is one of the cellular pathways that are set in motion using all of these transporters by changing the activity through activation and deactivation of certain transporters. In order for that to happen, in order for that to be possible, of course, we need these transporters here, these ATPases, that allow for the maintenance of this gradient, right? The membrane gradient, and the gradient between or across the tonoplast, that's this membrane here between the vacuole and the cytosol, is a different one. It's only 20 millivolt difference, minus plus, and minus plus here, 120 millivolts difference. So they're pumping much harder here to maintain that larger gradient than they're pumping, than even those two together are pumping here across the vacuole. All right, so let's sum this up a little bit, right? So that what I told you we have in summary form here for you to look up later and to go back to and see that you have the major points down. So vacuolar compartmentalization occurs via 3 hydrogen pumps that create an electrochemical potential that can be used for secondary transport. If you look here, you have the cytosolar hydrogen, so protons, and they can move in one direction into the apoplast, or they can move in the other direction into the vacuole. Hydrogen is pumped out of the cytosol. So that's one way strategy for ion exclusion from the cytoplasm, right? And the other one is the maintenance of the electrogenic gradient that I just pointed out to you across the tonoplast or across the plasma membrane. We have, as a third strategy, the activation of secondary antibody pumps, sodium versus hydrogen. I showed you that. I showed you the SOS1 as an example. I showed you the NHX1 in the vacuole. And I showed you the pathway by which sodium activates the calcium channel, which then activates the SOS3, SOS2 kinases, and the phosphorylation of SOS1, which then becomes active. So those are the 4 major strategies that the plant uses cellularly to combat salt stress. So in a nutshell, high sodium causes cytoplasmic enzyme inactivation. Here's some values for normal concentrations of potassium versus sodium. Sodium chloride in high concentration causes membrane potential imbalances. It changes the membrane permeability, so the membrane sort of becomes porous or brittle, doesn't really allow things to go through that used to be able to go through or not go through, and it decreases photosynthetic activity. Through mechanisms unknown, I say here, but of course, inactivation of enzymes that are part of the photosynthetic pathway are certainly part of that. All right, so that is it for today. So we will discuss on a particular example in the Gaxiola paper the strategies that I showed you here cellularly, and we'll look at examples of experiments that were done in order to understand this better, and in order to potentially create a plant that can be better withstanding salt stress. So this might be a potential way for agriculture to to produce plants that are allowing farmers to grow these plants even in saltier soils. All right, I will see you in class.